treatments for them. There are non-surgical treatments, there are surgical treatments, and none particularly work well. So he wanted to see, is there something that we could do, something that we could deliver into that femoral head, ball of the leg, and see if there's a way to prevent some of that destruction. And that's what we try to do, is in having a sort of minimally invasive approach to get the drug delivery system in there. It's a needle going through the side to inject, milliliter, less than a milliliter of this gel in there to see if, and so it was really the idea of trying to have a local treatment, not just have a kid take pills or inject something intravenously, but to have a local delivery that would try to control some of those adverse consequences. So you take the gelatin and you cross-link. Everybody's familiar with Jello. Everybody made Jello. So we just warm it up and it dissolves. We don't want that to simply dissolve because then the drug is just going to dump out. So we essentially cross-link it. So you make covalent bonds between the chains so that it doesn't really matter the temperature and within a physiological range at least, it's going to stay as an intact microsphere. And then we just treat those particles as sponges. So we dry them and then just let them absorb the drug-containing solution. So like taking a sponge, putting it in water, and they swell. They absorb water into it. It's just that when our gelatin microspheres swell, they pull drug in as well. Now what we didn't want is for the drug to just dump out when it is put into a physiological solution, because you have a concentration gradient that's just going to allow diffusion of the drug from the particles outside. So those different washing procedures, times, volumes, etc. were explored to try to get rid of the loose stuff, the loose drug, the drug that would burst out and give fairly high local concentrations before the body was really ready to start growing bone. So those wash protocols were to get rid of that burst, as we call it in drug delivery, to have a delay even before the gelatin particles, because they're protein, they will eventually be degraded by enzymes, and then the rest of the drug can start to come out over the next 3 weeks.
Okay.
Permanent implant. A drug delivery system that's going into an enclosed space, like the femoral head, is not going to be the same as putting it into, say, the abdomen. So it depends on where you need it. So if you have a location, a defect, where there will be some sort of constraint that the system can't, in this case, ooze out of, it's entrapped in that hemispherical head of the femur, that would be fine. In other cases, and many of our other systems, they are solids instead of gels. So it will contain the drugs, but it also, if you have a scaffold, it'll give a substrate, a network into which cells and tissue can grow.
Okay.
If we continue to wash, we could probably get rid of that little remaining burst, that whatever it is, 5, 10, whatever percent it is. Do we need it to be 0? Probably not. Would it be nice so it'll be prettier figure? Yes. But then you're wasting, in some cases, more drug. Because you have to think, when we wash out that drug, where is it going to go? Well, you think recycling, reuse, buzzwords these days, right? But when you're talking about biomedical products, when you're talking about drugs and biomaterials, you can't really reuse. You can't really recycle. So we could get rid of more, but then we are essentially throwing away more drug as well. So again, a balance. How low is low enough? We certainly didn't want 60%, that's too much. Getting into 40%, that's still more than we'd like, because in those initial phases after injection, the bone isn't really ready to start regenerating at that point. So we need to get control over the collapse that is in the early stages of occurring. You have collapse, then everything is gone. So that's why we wanted the anti-resorptive early, trying to stabilize the bone structure to some extent. And then after you give a few days for that to stabilize, then allow the drug, the simvastatin, the osteogenic, the bone formation promoting drug to start coming out. But we didn't necessarily need it to be 0, and we didn't want to remove some of the extra drug that would be coming out at a higher concentration later on by continuing to wash them. Okay. You know, where's the line between ex vivo, outside of life? So you have ex vivo, in vivo, and in vitro, right? So in an animal, which we did subsequently do, we have the in vitro, which is in the lab, but ex vivo is essentially in the lab as well. The one difference would be for some kinds of devices, cardiovascular devices, what they will do is sew in a vascular graft, for example, so that it will circulate from the animal through the graft and back into the animal. So the graft is essentially outside of life, outside of the animal, but it's still connected to the animal, period. So there's similar, we just used 2 different phrases. It's a good catch. We should have been more consistent, the legs out of the pigs, and just use them as a model to understand what will happen if we do try to inject this. Specialty of biomedical engineering, we're not in the business of trying to develop new drugs. Not to mention that's a huge process. That's a 10-year, probably $100 million project to get a new drug approved. And many of them aren't going to ever be approved, and that's why drugs cost so much, is because all that cost of developing them. So we don't want to develop new drugs. We want to use what is available, and that was the idea, is to take small molecule drugs that we could put into the system and deliver locally.
Yeah,
and it's not going to be a surgical procedure.
Right.
It's a needle going into bone, but you're not opening somebody up to do it. And then you have all that, so on the human side, the pain and rehab, but also the cost of putting someone in the hospital and all that. So it should be a comparatively simple procedure that seems to have some potential to work.